Hello, we're back once again. Get excited. Get excited for my new computer background. Get excited for the Final Fantasy VII remake that's already out. So let's take a look and let's pick up where we left off last time we were here. We were talking about biotechnology. And last time I asked you to watch a video about cloning the woolly mammoth and, talking about how close we are to doing that. I really want to know what you think of the video. Do you think the mammoth should be brought back, or what about other extinct animals? And if you do, why? And if you don't, why not? I would love to hear what your opinion is. So, moving on from cloning and that sort of use of DNA technology, let's discuss about a different use of DNA technology. So, why do you think the police would want to know someone's fingerprint? Probably to see if they were at a crime scene. So, if I stole this very valuable coffee mug, or if I was here rummaging around and I picked up this coffee mug to find the very valuable flash drive, then the police would know who I am because they could find the fingerprint, test them, and say, you're the one that touched this, so you were at the crime scene. Okay, well, what about a DNA fingerprint? Is that the same as the fingerprint on your finger? Nope. There's no DNA left from a fingerprint unless, like, you're cut and you leave some blood or your skin cells stay on there. Your fingerprint is an impression of the oils that are found on your skin that are left at a crime scene by touching something. And when you touch something like your phone, you can usually see here on here. I totally can see this giant fingerprint though. You can see that it's left. If you touch the inside of your glasses, don't do that. You can see a fingerprint. It's the oils that are from your skin left in the imprint of the ridges on your finger. And if you look at them, you'll see different like arches and swirls and whirls, and everyone's is different. A DNA fingerprint is not that. It's actually looking at your literal DNA that's left if you leave blood or skin or hair at a crime scene. And your DNA and mine is different, just like your fingerprint and mine is different. Our DNA is 99.9% the same. So how can they tell you from me then? Well, scientists will look at DNA that's different, the parts that are different. They know where to find these parts that are different between you and me, and that's what they look at. So how do you think scientists and police would use a DNA fingerprint? Well, again, just like using a real fingerprint, they would use that to find a criminal. If you've ever watched CSI or like Forensic Files and that, you might see they'll say, oh, the victim had, some skin under their fingernail, and they can sample that and get a DNA sample of whoever they were trying to fight against. Or if they leave like a drop of blood at the crime scene, they can swab it and send it and test it, and they can find out who was at the crime scene. That doesn't necessarily mean they committed the crime, but it does say that they were there at the crime scene. So this is a picture of an actual fingerprint. It's not mine. It's just showing you different things. So this is a real fingerprint, not a DNA fingerprint. A DNA or genetic fingerprint is a section of DNA that's unique to each person. That's the part that we use to tell the differences between us. Unlike your real fingerprint, scientists can use this to identify a person or an organism. This is what a DNA fingerprint looks like. These bars are sections of DNA. And what we do is we'll take some blood or some skin cells, we blend them up, and we get just the DNA out of those cells. Then they take that purified DNA and they squirt it into these little holes in something that looks like Jello. They actually call it a gel. It's like not that big. And then the Jello is like literally shocked with electricity. So electricity travels down the gel and the DNA moves through the gel. The gel you can think of like one of those big rubber band mazes that you'd have to like climb through and put your body through in all different ways. So big pieces of DNA get stuck at the top because it takes them a long time to kind of weave through this rubber band maze. And little tiny pieces go quickly to the bottom because they're kind of small and can fit through. Like I'm not a tiny person, so I'd maybe be somewhere near the middle. Like imagine John Cena trying to get through, even though you can't see him, trying to get through like a big rubber band maze. It might be hard for him because he's so large. For me, I'm just not in great shape. So it might take me a little while. Imagine like a small person that you know, or like a little kid. They could slip through easily because the holes aren't as big for them. So what you see here at the top, you see big pieces of DNA. Here we're getting smaller, 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 and these are the smallest. So how do you read that? Here's our crime scene. We have 3 suspects. We just look at, literally, all you have to do to read these, you look at where the bands match. And if you look from the crime scene in suspect 2, the colored bands match exactly. That means suspect 2 was at the crime scene. We found their DNA. Suspect 1 and 3, they might still be suspects, but their DNA is not at the crime scene. So how do we look at a DNA fingerprint? Like I told you, it's called gel electrophoresis, where they put the DNA, shock the gel with the DNA in it, and it travels down the gel. So DNA gets cut up by something called a restriction enzyme. Remember, enzymes build or break. The pieces are put into that gel. The gel is shocked, and the pieces move. The big pieces stay close to the top, and the little pieces go to the bottom. So here we have a mommy and their offspring, a baby. And then we have 3 dads. So this is a little bit like the Maury Show. Feel free to watch a clip if you've forgotten what the Maury Show is like. And we want to know which one of these 3 men is the father of this lady's baby. So this is a little bit different. We have to find the parent that matches up with the offspring. Okay, so it will have some in common with the mom. Like here's ones that it has in common with the mom. Okay, that one and that one. And then we have to find ones that it has in common with the dad. The dad is not going to have like it. You'll see. So like this one matches and this one matches with the mom and these match. Okay, these match. Okay, what about this one? This one matches. We have 1 in common. Then we have, let's see, 2 in common. What about this dad? We have 1, 2, 3 in common. And what about male dad 1? We have 1, 2, 3, 4, 5 in common. So male 1 is the father. There's the most in common with male 1. And the other parts that is from the child is from the mom. Okay, remember you're half mom and half dad. And that's how you use it to look at paternity. So let's look at some examples from a crime scene. Here's our crime scene sample. I like sound effects, by the way. Here's our victim. V is for victim. Okay, look, the victim's blood is not what's at the crime scene. They're different. We have 3 suspects. You want to know which suspect was at the crime scene. Pause for a second, see if you can figure it out yourself. Hit pause, like now. Okay, look, it's going to be suspect number 2. Do you see how the DNA from suspect 2 exactly matches the crime scene? Okay, that's how we know that the suspect 2 was at the crime scene. Now, this is evidence, I love this, from a real murder trial. This is one of the first murder trials where it was like kind of a big deal to use DNA evidence because it was discovered in the 80s. This trial happened in the 90s. I want you to ask yourself if you think this person's guilty. This is the standard. So that's just like a set of known DNA that they put in there to make sure the test is working, and it is. It's a little bit faded. I couldn't find a better picture. So these 3 are blood samples from the crime scene. You can see this one is different from crime scene sample 2, but crime scene sample 3 is the same as 1. You see darker bars, 1, 2, 3, 1, 2, 3. Here's 3, here's 3. It's really faded at the top, I realize, and it gets kind of ruined here at the bottom, but it's just a picture. So it looks like we have 2 victims, or at least we have 2 different bloods that we found at the crime scene. So this is victim 1. This was a double murder. Victim 1 is this blue one. Okay, so that's blood from the victim that we found at the crime scene. There's victim 2. That's her boyfriend that was killed. It's different from the other 2. So this is the 3rd blood type. It's not the blue one. It's not the red one either. So that means this is good for the prosecutors. The red one, that DNA is from someone else who was at the crime scene. So let's look at blood sample from the subject. Do you think that those match? You can look at them, take your time, pause the video, look at it as much as you want. Well, I hope you pause, because let me tell you who these people are. the red is, oops, O.J. Simpson. And then we have Nicole Brown and Ron Goldman. And if you know anything about this trial, O.J. Simpson was acquitted. He was found not guilty.